Welcome to my video on antigens. Antigen is a common term we use in immunology. And it is well known that anything that is foreign to the body is considered as an antigen. However, there are some self molecules that will act as the antigens, which are exceptions. In immunology, we also use some other terms like immunogens, haptens, epitopes, adjuvants, and we often get confused with these terms. So in this video, I will clearly describe what actually antigens are and how these antigens are related to or different from immunogens, haptens, epitopes, and adjuvants.
Let us see what antigens are. Our immune system has got the ability to distinguish between self and non-self molecules. Anything that is non-self or foreign to the body is considered as antigens. Once a foreign particle enters our body, then a battle begins between that antigen and the host immune system. The lymphocytes in our system produce specific immunological reactions against the antigens, leading to their removal from body, thus preventing the development of an infection or disease due to that particular antigen. These antigen or foreign bodies are usually bacteria, bacterial products, fungi, viruses, or other parasites. Entire organisms do not function as the antigen. Particular macromolecules of these infectious agents usually act as antigens. Proteins are the major antigens and polysaccharides ranking second. In contrast, lipids and nucleic acids of these infectious agents do not act as antigen unless they are coupled with proteins or polysaccharides. Antigens are also called antibody generators as they elicit an immune response by the production of antibodies. Antibodies, as we all know, are the glycoproteins produced in response to antigen by the host immune system, whose function is to facilitate the elimination of entered antigen. More scientifically, we can explain an antigen as any substance which, when introduced parenterally into the body, stimulates the production of antibodies and react with it specifically and in an observable manner. Here two terms are important: parenterally and specifically. To put up an immune response, antigen should be introduced parenterally. Parenteral means outside the intestinal tract. When antigens are given orally, they are usually denatured by the intestinal enzymes and their antigenicity is destroyed. So no antibody formation will take place. When given parenterally, they are not denatured, hence antibody production will take place. However, some antigens given as oral vaccines are exceptions. The next term I emphasized in the definition is specifically. Antigens are highly specific molecules. That means they will bind to only those immunocytes or the B or T lymphocytes which carry specific receptors or markers for that antigen. Antigens binding with lymphocytes may lead to either humoral or cell-mediated immunity. Binding of an antigen on B lymphocyte induces the differentiation into the plasma cells and the memory cells, followed by the production of antibodies, and thus lead to humoral type of immunity. Thus formed antibodies will react only with that particular antigen which had stimulated its production. However, cross reactions may occur between closely related antigens. On the other hand, the antigens that bind to T lymphocytes may not induce the direct production of antibodies. They will sensitize the lymphocytes, leading to cell-mediated immunity. So that was all about what antigens are. Now let us see what is the difference between an antigen and an immunogen. Immunogens are molecules that possess immunogenicity and immunological reactivity. Immunogenicity is the ability to induce antibody-mediated or cell-mediated immune response. An immunogen, when bound to B lymphocytes, induce the differentiation of plasma cells as well as memory cells. Antibodies will be produced by plasma cells, leading to humoral immune response. On the other hand, as already we mentioned in the previous slide, binding with T cells lead to the production of T effector cells and memory T cells, and thus set up cell-mediated immunity. The next attribute of an immunogen is immunological reactivity. Immunological reactivity is the specific reaction of antigen with the thus formed antibodies or the sensitized cells. Thus, an immunogen should be able to induce either cell-mediated immunity or antibody-mediated immunity, and should specifically react with the products of such immune response. In general, for all substances that induce a specific immune response, we use the term antigens, but more appropriately, they are called immunogens. Antigens are the molecules that possess antigenicity. Antigenicity is the ability to react specifically with the final products of humoral or cell-mediated immunity, that is either with the antibody or T cell receptor. That means antigens possess immunological reactivity but no immunogenicity. All molecules that have immunogenicity also have the property of antigenicity, but reverse is not always true. So we can say immunogens are molecules with immunogenicity and immunological reactivity, whereas antigens have only immunological reactivity, no immunogenicity. Hence, all immunogens are antigens. Now let us see what haptens are. In the last few slides, we have seen the difference between the term immunogens and antigens. But generally, we use the term antigen instead of immunogen. We usually say a complete antigen for an immunogen. So hereafter we use the term a complete antigen instead of an immunogen. A complete antigen should possess immunogenicity and immunological reactivity, otherwise called antigenicity. Means a complete antigen will induce the production of antibodies and specifically react with the thus formed antibodies. Haptens are incomplete antigens. Usually, complete antigens are large complex molecules that can induce an immune response by themselves. Haptens are low molecular weight substances. They are unable to induce immune response by themselves. However, they can react specifically with the antibodies. That means haptens are antigenic but not immunogenic. Then what is the difference between the antigen and a hapten? The difference is that a hapten can be converted into a complete antigen. The term hapten is derived from the Greek word hapten, which means to fasten. That means we can combine or attach haptens to suitable large molecular weight carriers, usually the protein carriers, and thus can make them immunogenic. In the given figure, you can see the small haptens, and when they are attached to suitable carrier molecule, they become complete antigen. This figure explains the effect of hapten on immune sera production. Here we can see that if the haptens are injected into lab animal as such, and if we observe the anti-sera production, there will be no change in the antibody titer. That means no immune response is generated by the hapten. If we combine the haptens to carrier molecules, as we already mentioned, like protein carrier molecules, and inject them to lab animals, considerable increase in antibody production can be observed. Means binding of haptens to suitable carrier molecules make them complete antigens and they become immunogenic. Penicillin is one example for hapten. Penicillin is not antigenic by itself. In sensitive individuals, it combines with certain serum proteins and thus initiate severe, sometimes fatal immune responses. There are two types of haptens: simple haptens and complex haptens. Simple haptens do not precipitate on reaction with antibodies. In other words, simple haptens are non-precipitating, whereas the complex haptens form precipitate on reaction with specific antibodies. The reason is that the simple haptens are univalent and the complex haptens are polyvalent. Thus, complex haptens have multiple binding sites for antibodies, which is responsible for the process of precipitation. That's all about haptens. Now coming to epitopes. Epitope is the smallest unit of antigenicity and it is also called antigenic determinant. In other words, it is the actual part of an antigen that is recognized and bound by antibodies, B cell receptor or T cell receptors. As mentioned before, usually the entire pathogen or the infectious agent will not act as the antigen. Protein or polysaccharide parts of these pathogens mainly act as antigens. Epitope is a small area on the antigenic part of a pathogen which will specifically bind to antibody, B cell receptor or the T cell receptors. Usually an epitope consists of 4 to 5 amino acid residues or monosaccharide residues and are immunogenically active region of an antigen. A single antigen can have multiple epitopes. Epitopes can occur in different conformations. Based on that, epitopes are of two types, namely sequential epitope and conformational epitope. When the peptide chain of epitope is occurring as a single linear segment, it is known as sequential epitope. It is also called linear epitope. When it is in a tertiary structure, it is called conformational epitope. In conformational epitope, surface residues are brought together from different sides by tertiary folding of the peptide chain. Usually, T cells bind to linear epitopes and B cells bind to conformational epitopes. Thus, epitope is that particular region of an antigen with a specific chemical structure, electric charge, and configuration capable of sensitizing the B or T lymphocytes and thus initiating the humoral or cell-mediated immune response. Similarly, on the antibody also, there is a specific part complementary to the epitope which will bind with the epitope. That specific region on the antibody corresponding to epitope is called the paratope. The diagram given here shows the epitope and paratope regions of antigen and antibody and their binding. Thus, epitopes and paratopes of the entire antigen and antibody are the regions that actually determine the specificity of immunological reactions. Coming to adjuvants. The term adjuvant is derived from a Latin word adjure, which means to help. So the main function of an adjuvant is to help the antigen to enhance its immunogenicity. Adjuvants are usually mixed and injected with antigens. Adjuvants are used to enhance antigens with poor immunogenicity or when the antigens are available in small amounts. Adjuvants are widely used in antibody production, for immunization, and also for research purposes. Adjuvants are known to promote the immunogenicity of an antigen in different ways. They can prolong the persistence of antigen in the body, enhance co-stimulatory signals, induce granuloma formation, also cause non-specific proliferation of lymphocytes. Friend's complete adjuvant is widely used one. It contains heat-killed mycobacteria in the water in oil emulsion. Alum and bacterial lipopolysaccharides are other examples of adjuvants in use.
Now let me summarize. So antigens are anything that is non-self to the body. The terms immunogens and antigens we use alternately, but actually the immunogens are the complete antigens with immunogenicity and immunological reactivity. Antigens have only immunological reactivity. Then haptens are the incomplete antigens which can be converted into complete antigen by binding with suitable carrier molecules. Then the next term epitope. Epitope is the exact part of the antigen which will specifically bind with the paratope of the antibody. Then adjuvants are the molecules that will help the immunogens to potentiate its immunogenicity. So I am winding up. See you soon with another topic. Till then, goodbye.